Hello everyone, welcome back to this chapter. Today let's see the second part. First let's talk about pollen allergies. Many of us have allergies towards pollen grains. Pollen grains you must have seen when you tap a flower on your hand, the yellow finite powder. These give allergies to many people. So it can cause severe allergies or bronchial pain. It can also lead to chronic respiratory disorders like asthma, bronchitis. Bronchitis is the inflammation of the bronchioles. The airway becomes blocked um, with phlegm, and the mucus is very thick. So it's very difficult to breathe in such cases. And one of the common examples here is Parthenium. Parthenium is very famous. Parthenium or carrot gas or gajar gas. It's also called Santa Maria. It's an alien weed. It's a weed actually which came along with the wheat which was imported from America. It is a contaminant actually because India at that time in 1950s there was starvation and the production was really low. So because of that, a law was passed and Congress government bought this type of wheat from America, imported it from America, and along with that wheat came this contaminant or this weed called Santa Maria, but it's called carrot grass because the leaves look just like the leaves of carrot. But these flowers produce pollen grains which are really highly allergic, also causes skin problems, and now it has become ubiquitous in India. It's just like a mess, and now so uh, scientists are trying to find out. I mean, beetles are being introduced which can kill this Parthenium, or other competitive plants are being introduced. So we just have to note that this plant produces pollen grains which causes allergy. Then coming back to pollen, yeah, pollen is also, it has a benefit, like it's sometimes taken as a supplement. It's a fashion nowadays to take varieties of food supplements. So because it is rich in nutrients, it's considered as a food supplement. You have tablets, syrups, and it's usually taken by athletes and race horses to improve their performance. And let's talk about pollen viability. Pollen should be viable from the anther to the stigma till they land onto the stigma. They should be viable. Then only it can lead to a successful fertilization or pollination. So pollens are viable because of their sporopollenin, which is the coating, which is the present in exine. But sometimes high temperature or humidity can affect the viability of the pollen. Apart from even UV radiations, or some biochemical or physiological changes can also affect pollen viability. Normally for cereals like rice and wheat, the viability is up to 30 minutes after they are shed from the anther. And whereas other plants, it can be up to months, like Rosaceae, medium-sized flowers like rose. Leguminosae means the legumes or the pulses we get from leguminous plants, or Solanaceae. So all these plants have the pollens have a viability of up to months. But for cereals, it's only up to about 30 minutes, but still it's a long time for a biological material to be viable. And pollens can also be made viable up to years if you keep them in cryobanks. Cryobanks, just like sperm banks, you have pollen cryobanks. So in India, you have it in ICARIIHR, Indian Council for Agricultural Research, Indian Institute of Horticultural Research. It is in Bangalore. And here, once you collect the pollen, you put it in this capsule like thing, and then you label it and store it. And you can store it in liquid. Here they are stored in liquid nitrogen. Sorry, this is degree. Yeah, liquid nitrogen. Nitrogen is a gas, we know. So it is liquefied and high pressure, and then so the temperature can be, it is liquefied, and the temperature of liquid nitrogen is 196 degrees Celsius, negative. Negative. So at that much cold temperature, if the pollens are stored, then it can be used later on. I mean, it can be stored for years, and then it can be used for uh, preserving some rare species or good varieties of pollen, and then it can be used for cultivation, breeding, etc. So these are tanks of liquid nitrogen where they are stored. Now let's go on to Gynooecium. That's the female reproductive part. In last video we had discussed about Androecium, the male reproductive part. So the female reproductive part, Gynooecium, it's also called pistil. Another common used term is carpel. Okay, so it can be a single pistil, just like if this is the ovary, and there are three parts. The first is stigma. This is called stigma. Then you have the style. Then you have the ovary. These are the main three parts of the female reproductive part of Gynooecium. So if it is just single stigma, single style, and this is called um, monocarpellary. It's called monocarpellary if it is single pistil. Pistil and carpel is almost the same meaning. So it's called single pistil or monocarpellary. If there are many pistils, then it's called multicarpellary. Here you can see three multicarpellary structures. I mean from three plants: hibiscus, papaver, and michelia. So here hibiscus, you can say that it has 1, 2, 3, 4, 5. It is pentacarpellaries. Instead of saying multicarpellary, since it's always 5, you can specifically say it as pentacarpellary. Carpel or pistil or Gynooecium is the same thing, okay? So here the carpels are fused. Here the carpels are fused. So in fused condition, it's called a syn carpus. And if here the carpels are free, so it's called apocarpus. And here also the carpels are free. Each is a carpel. Every carpel is free. So you can call it apocarpus. But here, since it is not, there are many, you have to call it multicarpellary. So based on the number, you can say them as monocarpellary or multicarpellary. Whether it is fused or free, based on that, you can say it's syncarpus or apocarpus. So this is michelia shows based on the number it is multicarpellary, based on the form it is apocarpellary. Papaver is actually poppy plant. Here I have shown poppy plant. Poppy plant is famous for opium poppy. You get opium and all the drugs from poppy plant. Poppy seeds are also used in cuisines. So papaver or poppy, it is fused, so it is apocarpus, and since it has many carpels, it is called multicarpellary also. And hibiscus, it is fused, so it's called apocarpus, and based on the number, it is called pentacarpellary. Now, if you look a typical flower, we are just going to study the single one, that is monocarpellary. It is easier to study. So you have a stigma, style, and ovary, and it is single stigma. Stigma is actually the landing platform for pollen grains. It is where the pollen grains land. It may come through air or by insects. But style is the elongated slender part beneath the stigma, just like in anther and filament, just like that. Here you have stigma and style. But here you also have the ovary. You also have the ovary. It's the basal bulged part of the pistil. And inside ovary you have ovarian cavity. You have the ovarian cavity. It's called locule. So we have to study the structures inside the ovary. And ovary is the part of the flower which later on develops into the fruit. Okay. If it is a vegetable or a fruit, it is the ovary that's going to develop into the fleshy fruit or vegetable. Okay, just to keep that in mind. And now let's study the inner structures inside the ovary. So this is the ovary. Inside you have the ovule. Ovule is also called megasporangium. So if this is the ovary, you have the ovule. This is also called megasporangium. So when you look into it, you can see that just like in human beings, there is a placenta. That's this dark, deep color, pink colored cells, which it has a lot of blood vessels. It provides nutrition to the fetus growing in the womb. So it is through this umbilical cord that the nutrients and gases are exchanged. But this placenta is enriched with blood supply, and it's part of the uterus which provides the same. An umbilical cord is the connection from placenta to the fetus. In the same way, just to have a link, I have shown this picture here also. If your ovary is something like the uterus and ovule is something like the baby, then here also you have a placenta and you have a connection. This connection is called a funicle. And where the funicle attaches to the ovule, it is called hilum or hilum. And when it is detached, you get a scar. Here in these seeds, you can see the scar here. These black eyes, these are the scars of the hilum. Just like the babies also have their navel or the umbilicus, in the same way these seeds also have these scars, and it's called hilum. So ovule is the small round structure here, and then this attached to the placenta by funicle, and then it fuses at the hilum. It's the junction between this ovule and the funicle, just like the baby and the umbilical cord. That junction is that in the belly you have the navel. So now we have to look into the detail, the structures, the coverings of the ovule, and what are the cells inside this ovule. So here, if we look at this ovule, you can see there is two coverings. This is the outer integument and this is the inner integument. And then you have a nutrient supplying cells called nucellus. It's not nucleus, it is nucellus. These are a mass of cells abundant in food reserves. And then inside you have the embryo sac. And inside that you have the egg cell. So this whole structure is not the egg. Actually, this is the ovule. Inside there are two coverings called outer integument, inner integument, the nutrient supplying cells called nucellus, and then you have the embryo sac. So this whole structure is called microsporangium or ovule. And here this embryo sac is formed. I'm sorry I've shown it directly, but actually initially there is just one cell here, and that cell is called microspore mother cell. This microspore mother cell develops into embryo sac and differentiates into these cells inside. So initially it's just one single cell here, that's microspore mother cell. So let's revise this once more. Integuments, these are the one or two protective envelopes around each ovule. They encircle the nutrient supplying cells called nucellus. Then there is a micropyle here. This is the opening through which the pollen tube enters. That is where the integument layer stops. There is a small opening, there's a gap here. That tip or opening is called micropyle. That's why when ovule has two poles, this is called the micropylar pole, and this opposite side is called the chalosal pole, and chalosal pole is considered the base. It's the opposite micropylar end, and it's the basal part of the ovule. And nucellus, as I said, is a mass of cells enclosed within the integuments, abundant in reserved food materials. An embryo sac, it is the actual female gametophyte. It is inside the nucellus, and it is formed from megaspore mother cell, MMC. Now let's see how this single megaspore mother cells develop into embryo sac. This is just one more picture showing the same thing. Here you can see this is the ovary, this is the ovule, this connection link, funicle, and there's placenta, stigma, style, micropylar end, and this is MMC megaspore mother cell that converts into embryo sac. So first let's look at how does this form. This is called megasporogenesis. Microspore mother cell from a single microspore mother cell. Here you can see these are the nutrient supplying nucellus, and inside this is microspore mother cell. The single cell undergoes meiosis to form two cells. So it's called dyad. Oh, I'm sorry, I'm talking microspore megacell. It's megaspore mother cell. Micro is for pollen grains. So now this is dyad. Now this dyad forms tetrad. Again, this is meiosis 1, meiosis 2, and it forms tetrad. We all know that meiosis gives 4 end products, and initially it is 2n, and these 4 cells will be haploid, single n. Out of this, 3 of them degenerate, and only 1 gets converted into embryo sac. So this megaspore mother cell, this is located near the micropylar end, and it is a large cell, dense cytoplasm, and a prominent nucleus. This process of these are now called megaspores. So it is formed from megaspore mother cell. So this process of formation of megaspores from megaspore mother cell is called megasporogenesis. The process of formation of megaspores from megaspore mother cell is called megasporogenesis. We had learned the same thing in pollen grains. It was microspores from microspore mother cell or pollen mother cell, microsporogenesis. Okay. Now let's see once more. This is the megaspore mother cell. It's a large cell located at the micropylar end and prominent nucleus. It is 2n. Then by meiosis, it gives 4 cells. 3 of them degenerate, and this is the one that goes to form the embryo sac. So it's also called monosporic development because embryo sac formation occurs from a single megaspore. Only one survives. While we were discussing about pollen grains, all the 4 microspores survived. Right. Now, let's see the embryo sac formation. So from that single megaspore, it has a nucleus. So this is haploid, and it has formed by meiosis. But now it is going to be mitosis. That is just duplication of the same content. Then only the nucleus divides. The cell doesn't divide, nucleus divides, and it gives two cells which goes to the two poles. Then again mitosis. This was also mitosis. Again mitosis, it duplicates itself. Only the nucleus divides again. So this is 4 cells. Now again mitosis. Each cell divides into 2. You get 4 cells and 4 cells. This is 8 cells. Now only the nucleus is dividing. The cell is the same single cell. So you should say 8 nuclei, 4 nuclei, 2 nuclei, but single cell. So it's called 2 nucleate embryo sac, 4 nucleate embryo sac, 8 nucleate embryo sac. These are the various stages. And now once 8 nuclei are formed, the cell wall starts forming. So only the nucleus divided, the cell did not divide, so it's called free nuclear division. Only the nucleus divided, not cell division. But once 8 nuclei are formed, cell walls are laid down and the cells organize themselves, arrange themselves. This arrangement is very important. 2 nuclei stays in the center, but 3 of them moves to the micropylar end and 3 of them moves to the chalosal end. The 3 moving to the chalosal end is called antipodal. This is the polar nuclei, and this is called egg nuclei, and these two are called synergids. So this is the arrangement of cells. When we look here, we can see that all the 6, 1, 2, 3, 4, 5, 6 cells have cell walls, but these 2 nuclei are lying in this central big cell. So this is called 7, 8 nuclei, but 7 cell stage. 1, 2, 3, 4, 5, 6 cells and the 7th big cell. Even though there are 8 nuclei, there are no 8 cells because cell walls are formed only around the 6 nuclei. And the central 2 polar nuclei lie in the central cell. So it's 7 cell and 8 nuclei stage. So if you look here, once more let's recap. Polar nuclei, it's this. It's 2 nuclei situated just below the egg apparatus. This is also called egg apparatus. Egg apparatus means it has 3 cells. It's towards the micropylar end. It has 2 synergids and 1 egg cell. And then what is filiform apparatus? Filiform apparatus is the thickening of the synergid cell. Cell walls, the cellular thickening at the micropylar end of the synergids. This is called filiform apparatus. It helps in the guiding the pollen tube into the synergid. The egg nuclei, it's very important. It is here, egg cell and egg nuclei. This is the one that gives rise to the embryo. The pollen tube comes in like this and the pollen nuclei fuses with this egg nuclei. So 6 nuclei are surrounded by cell walls. And antipodal cells, these are the T cells at the chalosal end. And the polar nuclei are located in the central cell. This is the central cell. So this is called 8 nucleate in 7 cell stage. Now next is pollination that we'll be dealing in the next video. Thank you.